not, let me know that you cannot see my screen. Okay, so welcome everybody, and thank you for joining this webinar. And again, as I mentioned right in the beginning, this is just a regular Zoom meeting. You have control over your microphone and camera, so feel free to turn them on if you want to, especially if you want to speak or say anything. Feel free to ask questions throughout. Don't worry about waiting till the end. Questions and suggestions and comments are welcome at any time. So just as a recap, we've both ViewPathDB and BVBRC, which I will describe those in a couple of minutes, have been running a series of webinars focusing on tick and tick-borne pathogens. And these are designed to be open community webinars and basically providing you some demos and showing you some of the key functionality of the resources. But also, we want to hear from you. And it's very important for us to know what data sets you think should be integrated, what functionality you need, and what tools you think should be built into these resources. So the webinar series, if you've attended some of them in the past, you would have probably met everybody on the screen, but we are at least the main presenters in this series. So Gloria Isabel Giraldo Calderon, based at the University of Notre Dame. Myself, I'm Omar Harb, based at the University of Pennsylvania. Anna Niewiadomska is at the J. Craig Venter Institute and is part of the BVBRC group, and Rebecca Wattam at the University of Virginia, also part of the BVBRC group. So let's see, making sure I admit everybody. Okay, good. Okay, so for today, what I'm going to do is I'm going to give you a quick intro on the Bioinformatic Resource Centers, or the BRCs. These are the national funded and supported mainly by the National Institute of Allergy and Infectious Diseases. I will remind you of this tick webinar series and show you where you can access previous recordings. I think there were some very nice tools that were demonstrated over the last few weeks and many more to come. And so definitely I would encourage you to review those. And then I will delve into some of the tools and functionalities that are provided by ViewPathDB specifically for eukaryotic pathogens transmitted by ticks. So focusing mainly on Babesia, we also have some data sets for Theileria, so I'll highlight those, but also describe additional resources that could be useful for you, including HostDB, Orthomcl, and our Galaxy resource for doing your own primary analysis. And then I'll leave some time in the end so we can make sure we have time to discuss and talk about see, I see there's enable the transcript. Sure, yeah, let me go ahead and enable the transcript. So you should start seeing some text popping up on your screen. Sometimes funny to watch it and see what it autocorrects, but let's see how it goes. All right, so I'll leave some time in the end for some open discussion, and obviously I'd love to hear from everyone. But again, this doesn't preclude you from turning on your mic at any moment and asking questions as needed. So the tick pathogen series is, as I mentioned, the ticks and tick-borne pathogen series. There've been we have 8 webinars, either have been conducted or a few more to go up until June. You can go and register by, you can scan this QR code, or you go to the link. My colleague Gloria will post the link in the chat. But also, and I'm going to switch quickly to the database. If you go to any of our databases, whether it is PyroPlasmaDB or if you go to BVBRC, for example, you will find an area where you can, you'll find links to the webinars. In the case of PyroPlasmaDB, if you go to learn how to use ViewPathDB, there's a section on webinars. And this includes not only the tick webinars, but also any previous webinars that we have run specifically for ViewPathDB resources. So there are lots of sort of 1-hour webinars throughout that have been conducted over the last couple of years that you may find useful. The tick webinar series has its own page right here, and you will see that the previous recordings are available, and so you can watch them. but also future webinars are listed here. Today's webinar obviously focusing on eukaryotic pathogens, and once this webinar is over later today, there will be a link here to the YouTube. I will also send an email to everybody who registered with a link to the YouTube, just so you you have access to it. Okay, so both BVBRC and ViewPathDB are one of two bioinformatic resource centers that are funded by mainly by NIAID. You're welcome to get more information or learn more about this mechanism by following this link or scanning the QR code. This resource, obviously, these resources were designed and they were conceived of to provide, and this is early on, so they started in 2004, it's been a while, but their goal is really to provide you, the researchers, with access to bioinformatic tools that allow you to access the genomes and access the functional data that's generated on those genomes, whether it is transcription data or metagenomic data or any kind of data that can be analyzed against genomes. Our goal is to provide you access to that and ways for you to query the data and reuse it. And I think that's one of our key goals, and that is to make sure that data that's generated over the years with great funding and great investment from different institutions, that data does not get buried somewhere in an archive or in the supplemental files of a paper, but rather make sure that they're available for you so you can reuse them and analyze them. And believe me, there's a lot of cool data out there that if accessible is highly useful and provides great opportunity to make some new discoveries on using old data, quote unquote. So as I mentioned, there are two bioinformatic resource centers that are funded. So ViewPathDB focuses on eukaryotic pathogens, vectors, and their hosts, and BVBRC focuses on bacterial and viral infectious diseases and their hosts. And our front pages look different, and that is because of the largely historical, because of the communities we serve, but also there are many differences between the types of data that are available and the ways that somebody working on bacterial genomes may want to access their data versus somebody working on a eukaryotic genome. And so there are some differences that necessitate some differences in the type of data that's available, the type of tools that are available. But we do work together, and our goal is to make sure that we synergize with certain tools, because there are some things that certainly overlap and we can really benefit from, and the community benefits in the end. So before I get into the demo, I wanted to highlight that there are many ways for you to communicate with us and reach out to us. So there is a form that Gloria will provide the link to it in the chat. And this is specifically for this TIC webinar series. So if you have specific comments or suggestions, definitely use that form. But if for some reason you're unable to access the form, you can go to any of our sites and click on the Contact Us link and send us an email. But also we are accessible through other means, through social media, Twitter, Facebook, and Reddit. Yes, we have a Reddit community, and you can go in there and type your posts or put up posts. And we also have a Gitter chat system installed on our resources. So in the footer of any of our databases, you can click on community chat, log in. You don't have to log in, unfortunately. It's too bad it's not an open thing. But once you log in, you can you type your question, and then somebody will get you. It's an open community chat, so anything you type in there is open. But there are a number of chats in there, and you may actually even find some answers to your questions in the trail of chats. Okay. So I'm going to now switch over back to PyroPlasmaDB, and I'm going to walk you through some searches and demos, just to give you a flavor of what is available in the resources and what can be done. And hopefully this will, hopefully teach you something new about what is you may want to try using these resources, and B, hopefully it'll get you interested in making sure that if there are missing data sets that would be useful to be integrated in, for example, PyroPlasmaDB, that you would let us know. We rely heavily on you, on the community, to let us know about data sets that are out there. It's almost impossible for us to track every single data set for the various resources that we support. So before delving into PyroPlasmaDB, I want to quickly hop to ViewPathDB. That's the parent resource. So all of the data that you find in all of our component databases. So looking here at the bottom of the page, you will see all these little cute icons. Each one of them represents a resource that's supported under the ViewPathDB umbrella. These include, for example, AmoebaDB or CryptoDB and PlasmoDB. You may be interested in PlasmoDB as well if you work on Babesia or Theileria, and of course PyroPlasmaDB, which supports Babesia and Theileria organisms. So, and all of these resources are built using the same infrastructure. This means that the resources all look the same. So if I jump to PlasmoDB, for example, I'm just clicking on the icon at the bottom of the screen. Let's see if I can do that. I'll do crypto. I think the live transcript is covering my screen here. So I just clicked on CryptoDB. You'll notice here what the page looks like. If I jump back to PyroPlasmaDB, you'll notice the page looks very similar, that the colors are different, the organism is different, and obviously the data content will be different. But how you interact with the data is exactly the same. And so the nice thing about this is that anything I show you today, you can apply this in PlasmoDB, for example, or ToxoDB. If you're interested in going to another apicomplexan and you want to leverage some of the data there, this is it's nice that all of them are built on the same infrastructure. Okay, so once you come to PyroPlasmaDB, typically you would expect that you have a question in mind. So you may want to visit a specific gene, or you're trying to search for specific types of proteins. And we have this option here at the top. It's a search bar, kind of similar to a Google search, I guess. Maybe not as sophisticated, but it definitely looks for the word that you type in here or the ID that you type in here in all the records that are available in PyroPlasmaDB. So for example, I can quickly type the word kinase and just search for anything that has the word kinase in it. And so this returns over 5,000 results right here. And you will see here on the left-hand side, there are a number of filters that I can apply on the results that are shown here in the central portion. So for example, you could see here quickly that there are, of these 5,000 or 5,100 results, there are about 4,000 that are genes. So these are genes that have the word kinase associated with them. There are a number of isolates that have the word kinase associated with them. And then there's some metabolic pathways, compounds, and some searches that have the word kinase in them. So you can use this to filter your results. So I can click on genes. And now all of a sudden, I'm only looking at the gene results. And I can now, you see here, I'm offered with additional filters. So I can, for example, say, well, I want only genes that have the word kinase that is in their product descriptions, for example. And so I can apply this. So I'm left with 774 genes. And then they may decide, well, I only want the Babesia microti gene. So I can just start typing microti here. This will filter my organisms. I can select microti and apply. And now I'm left with 80 genes in Babesia microti that have the word kinase associated with their product description, right? So this is a very specific search with specific filters applied on the search. I can decide that I'm done here, and this is great. I don't need anything else. I found my gene of interest, and you can go and visit that gene page and learn more about the gene. And we will visit the gene page in a few minutes. Or you can export this list of results into a search strategy. And so if I click on this link, this will basically take the 80 gene IDs, put them in the search strategy, and then you start. You're offered this workspace basically that allows you to build an in silico experiment. So you see this add step button here. This allows you to add additional experiments or additional analyses to your growing in silico experiment and ask questions about basically these 80 genes that I'm starting with. So for example, I can quickly say, well, do any of these kinases have a predicted secretory signal peptide? I don't know what the answer is, but I went ahead and clicked on the add step button. And what you will see here is that I now have this pop-up window, which tells me I'm going to combine with other genes. That's what I'm interested in. I can choose how I'm going to combine my results. In my case, I'm using an intersect because I'm interested in things that are both had the kinase in their product descriptions and also are potentially secreted. And now I have to select the search I want to intersect with. So in my case, I'm interested in secreted genes. So let's start typing secreted. And you'll see here a search that's offered to you is this predicted signal peptides. I could have, without typing in here, I could have tried to figure out, well, where would the signal, secreted protein search, where would that be under which category? And you might figure it out, say, oh, I bet you it's under protein target localization, and you would be right. Or it may not be intuitive right off the bat. And so being able to just filter these searches using this search filter bar here at the top is really nice. I use it all the time. I don't even try and look at the categories. And so I'm going to select this predicted signal peptides. And now I can select which organisms. I'm just going to select all because I'm doing an intersect, so it doesn't really matter. The intersect will only intersect with the right species, basically. I'm going to run the search. So this found 5,000 genes or 5,100 genes that are predicted to have a secretory signal peptide in all of the organisms in PyroPlasmaDB. Out of them, only one also had the word kinase associated with it, right? And so it was this particular gene. And then you can go and investigate it further and decide whether you believe that or whether it is potentially a mistake in the annotation or a mistake in the gene model, which obviously happens all the time. And so something that you want to make sure you take care of investigating. Okay, so I'm going to go back to the homepage. So I showed you this one search, which is the site search, which allows you to find things quickly using a keyword or a gene ID, and then you can build this in silico experiment. We're going to run a more sophisticated in silico experiment in a couple of minutes, but I quickly wanted to show you some other things on the homepage or that you can access from the menu bar here that could be useful to you. So one is, if you look under data, you can go to this page called Datasets in PyroPlasmaDB. And so this lists all the data sets that are available. Right now, there are 42 data sets. This includes genomes and other entities. And so the nice thing about this is that this allows you to then quickly see what is available. I see somebody raise their hand. If you have a question, feel free to unmute yourself and ask the question. Or if you prefer, you can also type it in the chat, whatever you prefer. Okay, I'll carry on. And you'll notice here, these are all the list of data sets that are available. And so you can, for example, start typing RNA in here, and anything that has the word RNA in it. I can continue going RNA-seq, and you'll notice here there are three data sets that are based on RNA sequence data that are available in the database, and you get information about them. So there's a Merozoite transcriptome from this Babesia species Xinjiang. And I will actually demo this data and take it a step further beyond just looking at this particular species of Babesia. And then there are a couple of Theileria data sets, and there are many more data sets that are coming in the pipeline. So anytime we hear about a data set, whether it is at a conference or from a publication, we keep track of it. And then as soon as it's available, we can then schedule it for a particular release. Okay, the other thing, obviously, that you may be interested in is looking for anything that is a genome. If you're trying to find out, well, which genomes are available in a database, so you can start typing again this filter here, and you'll see this category here for genomes. And so you can take a look at them and see what genomes are available. So that's one way of doing it. And then that'll give you sort of a good sort of synapse about what genomes are available. And so that's one way of doing it. I'll go back to the homepage here. And so another thing you can do is, if you're just interested in knowing, well, which organisms are in the database, you'll see here on the left, there's an organism search, and I can click on organism and genome info. And so this will run a search. It returns all the organisms that are in the resource. In this case, in PyroPlasmaDB, there are 18 organisms. And then you can scroll down here, and based on the information that's available in the columns, you can tell whether it has a gene annotation. And so you'll notice many of these genomes, these microti isolates, are not annotated, but they are assembled and they are available for you to search or blast against, for example. And then you'll see that other genomes have annotation. You can see the gene numbers. You can see if they're assembled into chromosomes. Some of them have gene IDs, but they're not assembled into chromosomes, right? They're just configs. So that just gives you an idea, and you can get a sort of a landscape analysis of what is the completeness of the various genomes. Again, between quotes, completeness, because none of them are truly complete. But that gives you an idea about that. Okay. And you'll notice here, this ran a new search strategy for organisms, and my old one is right here. But I can go ahead and close these. I don't really want them. And you'll notice I'm logged in. So if you log in, any search you run gets stored in your profile. And so that's one advantage of logging in. Although logging in is not required for you to run searches in PyroPlasmaDB, but it is required if you want to store your searches and make sure you can access them later on. Logging in is also required for some of the tools that are in PyroPlasmaDB. So for example, if you want to develop a workspace based on primary analyzed data, so using Galaxy, which I'll demo in a few minutes, you would have to log in because that's using storage and it's integrating, it's creating basically a private workspace for yourself. And so that requires logging in. Also, if you wanted to add a user comment on a gene page, that's all you need to log in because the user comment is associated with you, and I'll show you quickly how those user comments may be useful. Okay, so I'm going to go ahead and get started then with another search. And if you remember, when I went to the datasets, there was an RNA sequence dataset available for one of the Babesia organisms. So I'm going to go ahead and run a search based on the transcriptomic data. And you can access the searches in two ways. So one is on the left-hand panel here, you'll see there different categories. So I can search for genes. If I expand this, you will see all the searches available in different categories. These are the same categories that showed up at the pop-up that I showed you a few seconds ago. If you go under searches right here in this menu, you will see that all of these categories, the same ones that are on the left here, are also available here. You'll notice that there's a filter box for both of them. So it doesn't matter which way you get to the search. There are different ways to get to them. The nice thing about this banner or the header section is that it's everywhere on the site. So wherever you go on the site, this header section follows you, and so you can quickly jump to a search without having to come back to the home page. So that's a nice thing. So in my case, I'm interested in looking for an RNA sequence search. I'm just going to start typing RNA, and you'll notice here, oh, there's a search under transcriptomics for RNA-seq evidence. And so let's go ahead and select this. And you'll notice here, there's this Babesia Xinjiang data set from a Merozoite transcriptome. So that could be potentially interesting. So I can click on this percentile search. It's only available as a single type of search because there was only a single sample. And you'll notice that all of the data sets that I showed you here, all of them have single samples. So we can only do a percentile search. There's no comparison. But if we have multiple samples, then you can start doing comparisons and sort of looking for genes that are differentially regulated between different samples. Okay, so as I scroll down, you will notice here there's one sample. Then I can play around with the percentile expression, right? So this is ranking all the genes in this particular genome based on their expression levels, and from basically on a scale from 0 to 100, basically. And now I'm basically asking for any gene that is expressed above the 90th percentile, basically from 90 to 100% of all the genes that are expressed. So things that are highly expressed in the Merozoite transcriptome. And all the other things, you don't have to mess around with them because we only have a single sample, so nothing to worry about. You could, if you want, only look at all genes, so not only protein coding. So it depends on what you're asking. I typically leave it at the default. And now when I run the search, it'll return all the genes that had a that are expressed highly at the 90 to 100 percentile in this particular Babesia species, right? And so you can look down here, you can see what they are, what these genes are. You can look at the expression value. These are all, I'm getting back what I should be getting back based on the criteria that I selected. But you may wonder, well, you don't work on this particular species. What you work on is Babesia microti, for example, and so you really want to actually find the genes that are homologous to these genes that are highly expressed in these merozoids. So you could now leverage orthology to try and find genes that that are homologous to these genes that are highly expressed in merozoids of this Babesia xinjiang species. The biological concept here is that, well, if they're highly expressed in merozoids of that species of Babesia, then maybe they're also highly expressed in merozoids of Microtic, right? You can make these analogous comparisons. Of course, you have to confirm things in the end, but this is a nice way for you to do this in silico and sort of leverage data from one species and to another species. And so I'm going to add a step here. And rather than combining with other genes, I'm going to transform into related records, this option here. And this allows me then to transform my results into orthologs. And so I'm going to select the orthology option, the orthologs option. And now I can go in here and select specific species, or I can just select them all, right? And so I typically like to select them all and then, go back and modify things if I have to. And so now this will return these 309 orthologs had 3,699 orthologous genes across PyroPlasmaDB. And you can see they're all listed here. You can go and visit them individually, or you can apply some filtering. So you can see that Microti strain R1, there were 287 genes that are orthologous to these 309 genes. And so you can limit your results to just Babesia microti and apply this. And then now this will filter your results. You'll notice here there's a little filter icon, and then you can look at them here and examine them further, or you could leave the filter off. It really depends on what you're doing next. So what I'm going to do next is I'm going to add another step. And I do know that we have a proteomics data set for Babesia microti. And so I'm going to look for proteomics. So I'm just filtering on proteome, and you'll notice here there are different categories under proteomics, including post-translational modification, quantitative proteomics, and just mass spec evidence. So if they found the peptide, I'm just going to select the mass spec one for the purpose of demonstration. And I'm going to open up microti, and you'll notice here it's a Babesia microti whole blood proteome. And this is from Robert Molestina's group. And so I'll go ahead and select this organism, and I want to look for any gene that has at least one peptide. So basically, I'm saying that if it has one peptide from the whole blood sample, then that gene is made into a protein ultimately. And I'm going to intersect these results with my previous ones, right? So I'm saying basically, if this gene is an ortholog of the highly expressed merozoid genes from Xinjiang and is also has a peptide from the whole blood proteome, then maybe this is a really important gene that I would want to further analyze. So let's go ahead and run this step. And so you'll see here that there are 121 genes that were returned that are orthologs of the genes expressed in merozoids at the RNA-seq level, and also have at least one peptide that support its expression in whole blood samples from Babesia microti. All right. So you may decide that you're done here, and this is your big list of 121 genes that you are going to explore further, or you may decide to start dividing this up into subgroups based on what features these genes have. So, for example, if I'm looking at things that are expressed in merozoids and whole blood, I might be interested in things that are potentially secreted and maybe are at the interface between the merozoids and the blood cells, and it's because I'm interested in maybe designing or finding a new potential vaccine candidate. So what I can do is I can add another step here, and again, just as I was doing before, I'm adding a step, and I'm going to look for more genes. And remember, I ran the secreted gene or secreted protein analysis. So I'm gonna, or search. So I'm just going to do the same thing. I'm going to run search for predicted genes that have a predicted secreted signal peptide. I'll select all again, and I'm going to run the search. And then you'll notice here that now I'm left with 5 genes. So these are 5 genes that have at least one peptide from whole blood samples that are orthologs of Babesia xinjiang genes that are highly expressed in merozoids. And then ultimately, as a biologist, you want to go down and look at these genes and see if they make sense, right? And some of them may make sense, and some of them may not make sense, right? And so we can explore these further. I'm just looking at these to see if there's anything interesting here. So this phosphatase may be interesting. These are heat shock proteins, so these are things you may want to explore further. So let's go ahead and look at this phosphatase. So I'm just going to click on the ID, and this will take me to the gene page for this particular phosphatase. And so now we can scroll down and look at it further. You'll see here, this is this gene right here. I can look at the gene model on the gene page and zoom in a little bit. We see here I had to activate the scrolling, but once you activate that, you can zoom in. You can also open this in the browser, and then you have much more functionality there where you can add any data tracks. But you'll notice this gene has 1, 2, 3, 4 exons, and it's going from right to left relative to the orientation of the chromosome. And so that's potentially interesting. So let's keep track of this. And you'll see here, this gene page will go down, a few floors down to the basement, but there's a lot of information on here. And the more data we have, the more information there is here. You can either scroll down slowly down here and examine everything, or you can use these categories to jump around. So for example, I'm curious to see whether this gene has orthologs in other Babesia, and based on the search I ran, the answer should be yes, because it's an ortholog of all of them. So, and I clicked on this orthology and synteny section, and you'll notice here that it does have orthologs in all the organisms in PyroPlasma-DB. And so that's interesting. It seems to be conserved across all Babesia. I can go down and examine other sections, but what I'm going to do is I'm going to hop onto Orthomcl, which is the resource that we use and we develop, but we also use it to define these orthology clusters. And so basically, Orthomcl contains over 500 genomes from across the tree of life, and all the proteins from these genomes are blasted against each other, and then based on their similarity and alignment, they go through a clustering algorithm which then puts them into groups. So anything that falls into the same group is considered an ortholog of each other. And so all of these genes fell into the same orthology group. That's why they're considered orthologs of each other. And this orthology group will contain, it could be limited to just Babesia and Theileria, but it may also have other members from other organisms across the tree of life, including other alveolates, for example. So I'm going to go ahead and go to Orthomcl by clicking on this link. And so this is going to take us to this other database that we maintain. And you can quickly go down here. This is the orthology group for this particular group. This gives you sort of a phylogenetic tree describing where this gene is present across the various organisms. And I can quickly look at the alveolates. You can see it's conserved in the free-living alveolates like Chimera and Vitrella. And then if I go down to the other, Apicomplexa, you can see that it is also present in Plasmodium. It is conserved in the Pyroplasmida as expected. We could look at the Coccidia, and you'll see that it's also conserved across all of them. In fact, it looks like they have potential paralogs or it's duplicated in the other organisms, and so that's interesting. So we can go down now and look and see, well, what are these proteins in all the other organisms? And so let's go ahead and look at this. And I notice that it has an ortholog in Toxoplasma, and Toxoplasma has been heavily studied for proteins. We could look at Plasmodium as well if we want, but I'll go ahead and look at Toxoplasma. And you'll notice here that it does have an orthology to this acid phosphatase, a GAP50. And so this looks interesting. I know that GAP50 is involved in potential motility of the tachyzoid stage. So that's kind of interesting, and one would want to go and examine this further. And so you can do this by either looking at it here in Orthomcl, and this will take you to the protein page for this Toxoplasma gene. And now I'm going to jump to ToxoDB. All right, so this is another database, and it seems like we're jumping all around, but remember, I'm looking for this potentially really interesting protein that I found in my search in PyroPlasma-DB. And now I'm going to look at the Toxoplasma ortholog to see if there's more information about this in Toxoplasma that may then give me some hints. Is this an important gene? Do I want to investigate it further? And the first thing, obviously on the gene page, it looks the same as the PyroPlasma-DB gene page. It's the same structure and everything. One thing you'll notice here is that there are 2 user comments available on this gene. So whenever I see user comments on a gene, that's pretty cool, because it tells me that somebody out there who uses the resource added a comment on the gene, and this could be potentially useful. And so once you click on that, it takes you to the user comment page, and you'll see here that there are 2 comments. One is from Stuart Ralph at the University of Melbourne, and one is from David Bruce here at UPenn. Both work on Toxoplasma and Plasmodium, so that's pretty cool. I can look at these comments directly, and you will see here that Stuart Ralph indicated that this is an acid phosphatase, and has some information about it, which is nice. And you can always email them directly and say, hey, I noticed you put this comment on. Can you tell me more about why you think that? And then David added a bunch of papers which show you that this is an important gene involved in the myosin complex. And so now we can start building a story about this gene in Pyroplasma-DB. So I'm going to go back to Pyroplasma-DB here. So this gene, which is potentially secreted based on the signal peptide algorithm, it's highly expressed in merozoites of another species, but also has a peptide from microti in whole blood cells. And its ortholog in Toxoplasma, we know, is important and involved in invasion and motility. So that's pretty cool. And so now you have potentially a protein you would want to investigate further if you were interested in these types of proteins. So I will go back to the strategy. I want to make sure we leave some time in the end for some questions. So one thing I wanted to show you here, and I noticed this when I was looking at this gene, which I thought was kind of interesting. I'm going to go back to the orthology and synteny section. And now when we look at this section, you'll notice these were all the orthologs, which I showed you before. Let me collapse this section here. And we have a section for synteny. This is basically an alignment of all the genomes in Pyroplasma-DB, or most of them at least, showing you here in this window. And then the orthology is the shading shows you where the orthologs are. So I'm going to, let me enable scrolling. I'm going to zoom in a little bit. Okay, so we have, this is the acid. I can click on it and get more information, just confirm that this is the acid phosphatase. It's the right gene ID 05790, which is the one right here. So that's good. I'm looking at the right gene. It has an ortholog in other Babesia and also in Theileria. But one thing I noticed right away is that in the other organisms, it only has 2 exons. Whereas if you remember, in Babesia microti, it had 4 exons. And so this is interesting. So either there's something really special about microti where this gene has more introns for whatever reason, or there's a problem with the annotation, right? And so that's where having some RNA sequence for microti would be incredibly useful, and so hopefully we can load some in the future. But remember, for this Babesia Xinjiang, we had the RNA sequence data. And so you can take a look at it there and see if you believe this, the RNA sequence data, and if it does give you any support. And so I'm just going to go back to here. So I loaded a gene, just preloaded this gene page to make it easier. And I'll go ahead and scroll down here on the gene model page. So remember, in all the other Babesia organisms, it was a 2 exon gene. And then if we scroll down here, we have a track where we look for reads that go over, that have to be split when you map them back to the genome, which means they have evidence for introns. And so you'll notice here for this gene, there is one, or there are over 2,000 reads that go over one section, indicating that there's only one intron in this gene. So the evidence for this being a single intron gene or a 2 exon gene in Babesia Xinjiang is really strong, right? And so you can be confident that it's probably a 2 exon gene there. And so that's interesting. And I would, if I was going to work on this gene in Babesia microti, I would make sure to confirm the gene model before I start cloning, because there are problems with the gene models, and the underlying data can really give you support or not for that particular gene model. So quickly, I'm going to go back to my searches, and just to highlight that there are other things you can do, other types of searches that are available. So for example, I'm going to go ahead and delete this step, the last step, the signal peptide. And I'm going to, for microti, I know we have a number of isolates from different studies that are available and we use them to align them back to the reference genome to call variants. And so you can start asking, well, are any of these genes, these 121 genes, are any of them under any kind of selection? Do they have a high number of non-synonymous SNPs, for example, SNPs that are causing a change in the amino acid sequence? And this would tell you maybe those are genes that are really at the interface with the immune system because they're under pressure. And so let's go ahead and look for SNPs, or you can look for variation. So there's a search for genes based on SNP characteristics. And again, I won't go through all the details of this particular query because it we have entire webinars that are dedicated just to the search, and I would encourage you to go and view them. But you can select all the samples that you're interested in. In my case, I'm going to select the 49 samples that are available for my CRODI, and then I can play around with the different parameters. I'm looking for non-synonymous SNPs, and I'm going to just say, well, I want any gene that has at least 10 non-synonymous SNPs. I'm basically arbitrarily deciding that that's a high number of SNPs for my genes of interest. I'm going to increase the stringency here a little bit and change it to 60. And again, there's a lot of information here for each of these parameters. So definitely play around with this, or send us an email if you have questions about how to run the search, and we're happy to do it. And so when I ran the search, it found 965 genes that met the criteria, and only 2 of them were part of my 121, right? So they're genes that met all the previous criteria, and they have at least 10 non-synonymous SNPs in them. And so then you can take a look at them and decide maybe these are these are genes that are maybe under pressure to change, maybe they're interacting with the system or not. And so that'll be kind of curious. This one hypothetical, which would be very interesting to look at, and this ATPase, which is also interesting to look at. Okay, I'm going to go back one step here. So I'm going to click on this 121 genes, and I want to show you a couple of additional analyses that you can do. So once you have a list of genes, and this list of genes can come from a search strategy that you're running here based on data that's available, or I can run a new search and I can upload a list of genes. So you may have your own list of IDs because you ran your own proteomics experiment and you've decided, oh, these are the 200 genes that are really important from whatever organism that's available in PyroPlasmaDB, and you can run this search and just paste your IDs in there, and then it'll start a strategy like this, and then you can integrate it and ask questions about it. So one of the more popular searches that people run with their own lists of genes is they can do 2 things. One is they can do a genome view. This shows you the distribution of the genes. So each one of these ticks indicates a location of a gene on the chromosomes or the contigs, depending on what they are. And you can see that chromosome 4 had 44 of those genes, and you can, get a really rough estimation of whether there's any kind of clustering going on or anything interesting. And then you can also analyze the results, and you can do this by different ways. These are enrichment analyses, statistical enrichment. And so, for example, you can run a Go enrichment. If I click on Go here and I run, let's say, I'll just keep the defaults for now and submit this. This is going to determine if my 121 genes had any Go terms that were enriched in this 121 compared to the background, which is the genome. I don't know what the answer is, but we can take a look here. It looks like some broad categories like translation had pretty high corrected p-values, and as we scroll down, you can get a bit more specific Go terms that may be of interest and to explore further. Okay. So. And again, with any of these steps, you can modify them, revise them. You can save a strategy. So if you're logged in, you can save this. So I can, for example, go ahead and save this and say webinar strategy, save it. And now once you save it, you can generate a unique link. And this unique link now I can copy it. And if I can find the chat window, I'll go ahead and send it to everyone in the meeting. And so now I put this in the chat window. If you click on it, it's going to open the same search that I ran here, but in your own workspace. So then you can play around with it and do whatever you want with it. It's a really nice way for you to interact with your colleagues and send strategies around. Okay, so I mentioned to you that we leverage Orthomcl. And so I'm going to go ahead and delete this last step. And so we leverage Orthomcl in 2 ways. So one, it allows you to use this transformation step, which I showed you earlier, which allows you to look for orthologs of a list of genes in other organisms in the database. Another thing you can do is you can find genes that meet a particular phylogenetic profile. And so for it, I can add a step here, and I can, again, I'm going to look for genes. I'm going to intersect my results, and I'm going to look for phylogenetic profile. And you'll notice here that there's an orthology phylogenetic profile search. And now I can I'll select all of the organisms in PyroPlasmaDB. And now you can define, well, what kind of phylogenetic profile are you interested in? And what this search will do is it will, for example, if I go in here and say, I want to find any gene from any organism in PyroPlasmaDB that does not have any homologs in mammals, for example. So I can go under core data and you'll see mammals. And now I can click twice on this little dot here. So it puts an X here. So this is defining the orthology group. The orthology group is going to be any group that does not contain mammals. And so if it doesn't contain mammals and contains one of the organisms in PyroPlasmaDB, I want to return that gene so I can just find out if any of my 121 genes here are part of that gene list, basically. And so now I've defined this. I'm going to run the search. So this is going to look for all the orthology groups. So there were 25,000 that did not contain mammals, but had genes that are available in PyroPlasmaDB. 24 of them were part of those 121, right? So now you can go down and take a look at them. So these are presumably, if you were going to use any of these for developing a vaccine, obviously any of these, or use them as an antigen in an experiment in a mammal, for example. So any of these that do not have a homolog in mammals may be more attractive because you won't generate self antibodies or self response. And so that's kind of nice. And then you can take a look at these and explore them further. Okay, so I'm going to show you one more thing, and then I will open the floor for discussion. So I wanted to highlight the Galaxy workspace. So if you go under tools in any of our resources, you'll notice here you can go to Galaxy. And what this will do is it'll open up a page that looks like this. You have to log in because this is going to create your own personal workspace in Galaxy. And then you'll notice here that we have a number of, and if you're not familiar with Galaxy, there's a lot of, we don't develop Galaxy. It's an amazing, amazing web interface that allows people like you and me who are maybe not computational biologists to access all these informatics tools using a web interface. And you can build these really nice workflows, just like you would do a workflow in the lab where you're running an enzymatic digest, for example. You're doing a workflow. You take some DNA, you add some buffer to it, you add some enzymes to it, you put it in an incubator, you come back in an hour, you've purified the DNA and so forth. That's a workflow, right? And you can generate a workflow which is composed of bioinformatic tools. And you can then run your own RNA sequence analysis or variant calling, or you can map your genes to Orthomcl, for example, to assign them to groups. This is an example of what I did here. I took a list of sample proteins. These are just in FASTA format, and I mapped them to Orthomcl, and then you come back with lists of genes that are potentially interesting and that you may want to explore further, and then you can download these and work with them. And then there, as I mentioned, there are a number of RNA-seq workflows. They're under different categories. But let's say you wanted to run them, if you had, let's say, data available, you can upload your FASTQ files, which are the reads, and then you can run any of these workflows. So, for example, this is a paired end unstranded, and so if I click on this, this will open up the interface, which allows you to select the samples that you want to compare to each other, and then the different parameters here that you can play around with. It takes a while to get used to this, but ultimately you can run your workflow and then generate some primary data that you can use and import back into PyroPlasmaDB and investigate further. And we're more than happy to cover these with you in more detail if you're interested. And we also have a number of additional webinars for you to explore. Okay, so I'm gonna stop here and open the floor up for questions. So let's see who is still online here. So yeah, so let me know if you have any questions or comments. In particular, obviously, if you have questions about anything I demonstrated today, I'm happy to go into it in a bit more detail. If you're aware of data sets you would like to alert us about, this is a nice opportunity. Obviously, not the only opportunity, but this is one opportunity for you to do that. Or if there are any tools or things you think are missing in the resource that you would like to hear about. And I'll be quiet for a few seconds to give you an opportunity to unmute yourself and speak.
Hey, Omar.
Hey.
Very nice. I'm always amazed at what you guys have pulled together here. One type of data that might be useful, I don't know if you've considered this, and maybe you have, because it seemed like in the one example that you used, David had pulled in a bunch of papers. But have you considered linking to publications where specific genes have been modified, have been undergone genetic manipulation? Because that type of sort of orthogonal data could actually be useful in interpreting the significance of individual genes.
Yeah, no, that's a really good point. So we do have ways of linking to papers. That's straightforward. I think the harder part is pulling out the information from the paper to indicate that this is a particular mutation and that's causing a particular phenotype. So so one thing that we haven't implemented yet, but what we've thought about doing, and that is allowing users. So for example, if I go back to my search strategies and let's just pick any gene here, you can obviously add a user comment, right? Anybody on this call, if you just create a login, you can add a user comment on a gene page, and those are, the barrier is very low to add a user comment, and that's on purpose because we didn't want this to be arduous on people. But you can, indicate whether it's a phenotype if you want, and you can link a paper, right? So I can, if I just I'm just adding a random number, and then you can decide if this is a real number, and then link it to the gene. But we are also considering developing one specifically for phenotypes, and so a specific form that is a bit more structured, that allows you to then specify exactly what kind of genetic mutation was there, and then how, and describe it, using sort of an ontology that makes it structured and easier to then allow people to search on that kind of data.
Okay.
But yeah, no, that's a really good point. And the automated ways of sort of text mining, they could work for pulling out some genes, but then it'll be hard to then associate with what information was in the paper that then makes it useful. Yeah.
Well, even this is, I think is probably, I didn't realize this was in there, but that'd be really helpful.
Yeah. And you can actually search for, so we can do the search right now. I don't know how many comments are in ViewPathDB, but we can do a search for user comments.
Right,
you'll see there are not that many, but they're 159, which is actually not that bad, So and so you can take a look at them then and see, and you can see the comment headline, right? And so now this gives you a quick idea of what people have been saying about these genes, and that's pretty cool. And sometimes, this gene here, whoops, bounced around, let's see. So yeah, it's possible that in some cases, in many cases, it'll be telling you something about the function of the gene, right? Which is quite nice. Like in this case, whoever added this comment is saying that these are perforin-like protein, and so that will be quite useful if you're working on this gene and it's annotated as a hypothetical. I don't know, let's take a look at one of these. It says MAC perforin, so it had an idea in there, but then this person who added the comment, where is it? Bounced around. User comment, there we are. Oh, my page is jumping around, there we go. Martina Paoletta, she added this, and so then you can decide, whether you're going to trust this or not, but there's a paper associated with it, so this is really cool, right? So then you can actually go in and be a bit more confident about this.
Yeah, okay, great. Yeah. Thank you.
Sure. Yeah. And then thank you for joining, David. So again, while people maybe are thinking about questions, I wanted to highlight one more thing that you can do if you go back to the parent portal, ViewPathDB, because I love this idea of being able to leverage data from other organisms, because there is some information there that could be quite useful. And I'm going to just highlight one search. I'm going to look for this CRISPR search. We have there's a whole genome CRISPR mutagenesis in toxoplasma, right? And so you may argue that, oh, if a gene is essential in toxoplasma, right? So I'm going to look for anything that's, and there's information here which you can learn more about the particular data set. But I'm going to look for any gene in toxoplasma that was considered essential based on this data set, and there are 368 genes that met this criteria. But if I work on Babesia, you may be interested in knowing, well, if I have an ortholog in Babesia, maybe it's also essential in Babesia, right? So you can use this as a way, again, as a surrogate to look for potentially essential genes in Babesia. So I'm going to transform this by orthology, look for orthologs, and in this case, I'm going to look for, let's search for microti. And so let's just transform to microti orthologs, or you could do it more. And it looks like there are 275 genes that are orthologs of these 368 genes in toxoplasma that are considered essential or important for fitness, right? And so then you can go in and take them, and that's a way for you to then narrow down. Maybe if you have a large list of genes, this may allow you to narrow down which specific genes you want to maybe target first or work on first, because there's some information from other organisms. So this is a really nice way to leverage data across sort of related organisms. All right, any other questions? Or again, maybe I'll put up the PowerPoint again. Can find it. There we go. And let's go back here and just remind you that if you have,
we cannot see the PowerPoint, Omar, just ViewPath.
Oh, really? Interesting. I wonder why. Let me stop sharing. Share again. How about now?
Yes. Okay,
perfect. So, so there are different ways of contacting us. We do have a form specifically for this webinar series. If you prefer to do this, you can be completely anonymous, or you can add your information. We also have a very active support email list. We love hearing from you. So anytime you have a question, if you are not sure about how to run a search, or if you just want to tell us about something that's not working right, or suggestions for additional data sets, drop us an email. If you're active on social media, definitely tag us, and we're happy to hear from you that way as well. And we also have on all of our pages, we have a community chat, which you can click on and start chatting. You do have to log in for the community chat, which is because we're using Gitter, which is a third-party open community forum. Great. Well, if there aren't any other questions, then thank you everybody for attending, and hopefully you found this useful. As I mentioned, the recording will be made available, and we will put it on our webinar page, and also we'll send an email to everybody who attended this, who registered for this webinar, including yourselves, obviously, with a link to the recording. Thanks again, everybody. I appreciate you joining, and hopefully we will see you somewhere soon in real life or on another virtual webinar. Bye, everybody.